Welcome. Today, we're diving into the world of sterilization in place. We'll explore the core principles behind it, look at where it's applied, and understand the regulatory landscape that shapes its use. Let's get started with Dr. S.K. Singh's insights on this essential topic. Let's start by understanding why sterility matters so much in pharmaceuticals. For products that go directly into the body, like injections, eye drops, or inhalers, keeping them free from microbes is absolutely critical. That's where SIP, or steam in place, comes in. It eliminates contamination, prevents spoilage, and keeps everything aseptic. This not only protects patients, but also helps companies meet strict regulations. And the industry is evolving, moving from manual sterilization to automated, then smart systems, all aiming for continuous sterility assurance. Let's talk about sterilization in place, or SIP. It's a carefully validated process that sterilizes equipment and systems without taking them apart. Instead, it uses saturated steam or other agents to kill any contaminants inside. The main goal here is to reach a specific sterility assurance level, making sure everything is reliably sterile throughout the entire system. You'll find SIP used in places like bioreactors, fermenters, formulation vessels, transfer lines, holding tanks, and even the product pathways in filling machines. It's all about keeping things clean, efficient, and safe. Let's take a look at the regulatory framework guiding our work. Several key organizations set the standards we follow, from the U.S. Food and Drug Administration to the European Commission, and global bodies like the World Health Organization. Industry groups like the International Council for Harmonisation and the Parenteral Drug Association also play a big role. The bottom line: SIP systems need to be carefully designed, validated, and regularly checked to make sure sterilization is always reliable. Our goal here is clear: to achieve complete sterilization throughout the system. We rely on specific conditions: temperatures between 121 and 134 degrees Celsius, pressures from 15 to 30 psi, and exposure times lasting 15 to 60 minutes. The key principle is using moist heat to destroy all microorganisms and bacterial spores. This works by denaturing proteins, destroying enzymes, damaging cellular membranes, and inactivating spores. These combined effects ensure thorough sterilization every time. Let's talk about sterility assurance level, or SAL. It's a way to measure how likely it is that a single microorganism survives after sterilization. The industry standard is set at 1 in a million. That's SAL 10 to the minus 6. In other words, out of a million sterilized units, only one might still have a living microbe. Regulatory bodies require validated sterilization systems to consistently meet this level. This gives us a clear, measurable assurance that sterilization is truly effective. Let's take a closer look at the key components that make up a typical SIP system. You've got the steam generator, which creates the steam. Then there's the sterile steam distribution network, pressure control valves, temperature sensors, condensate traps, vent filters, and the automated control system, usually a PLC or SCADA setup. But it's not just about parts. The design has to be hygienic, minimize dead legs where steam can stagnate, ensure complete steam penetration, and remove condensate effectively. These requirements are critical to keeping the system reliable and safe. Let's take a look at the typical SIP cycle, that's steam in place, and how it keeps equipment sterile. It starts with cleaning, or CIP, which must be done and verified before anything else. Then we move through preheating, air removal, and steam introduction. After that comes the sterilization hold phase, where everything is held at the right temperature. Finally, controlled cooling, sterility maintenance, and equipment release wrap up the process. This sequence ensures safety and compliance every step of the way. When it comes to regulatory focus, we're talking about keeping a close eye on the critical process parameters that ensure safety and effectiveness. These include temperature, which drives microbial kill efficiency, and exposure time, essential for sterility assurance. Steam quality matters too. It affects heat transfer. Pressure helps maintain saturated steam conditions, while condensate removal and air elimination prevent cold spots and ensure uniform sterilization. Each parameter requires careful validation to meet strict standards and keep processes reliable. When we talk about SIP validation, our main goal is clear: to prove that the sterilization process reliably achieves sterility every time. This involves several key stages. First, design qualification confirms the equipment is suitable for the job. Then, installation qualification ensures it's set up correctly. Operational qualification checks that everything runs within the right parameters. Finally, performance qualification demonstrates consistent sterilization during routine use. Throughout this process, thorough documentation is essential. Detailed validation reports keep everything transparent and accountable. Let's talk about biological indicators, a key tool for validating sterilization processes. One commonly used organism is GEOBACILLUS STEAROTCHRMOPHILUS. It's highly heat-resistant, which makes it perfect for testing. We start with a known population, usually 1 million spores. After running a validated sterilization cycle, no growth is observed. That means the sterilization was successful. It worked as intended, ensuring safety and reliability. Let's look at the validation results for the sterilization in place cycle. Every temperature probe hit the target temperature, confirming uniform heat distribution. Biological indicators came back negative, so no contamination survived. We also found no cold spots anywhere in the system. With these results, the SIP cycle is approved for commercial manufacturing. This example used a 2,000-liter stainless steel bioreactor, running at 121 degrees Celsius, held for 30 minutes under 18 psi pressure. This validation ensures consistent, reliable sterilization every time. When regulations aren't met, the consequences can be serious. We're talking about everything from GMP observations and warning letters to product recalls and even production shutdowns. Common issues often show up during inspections, things like inadequate temperature mapping, poor steam quality checks, or dead-leg design problems. Incomplete biological indicator studies and weak requalification programs also come up a lot. Missing alarm management procedures and poor data integrity controls round out the list. These gaps can quickly turn into costly setbacks. Looking ahead, SIP technology is evolving fast. We're seeing smart SIP systems that adapt and learn. Real-time steam quality monitoring ensures every cycle hits the mark. Digital twins create virtual replicas, letting us test and optimize without risk. AI steps in for cycle optimization and automated deviation management, catching issues before they happen. Predictive maintenance keeps equipment running smoothly. Continuous process verification and Pharma 4 integration bring even more precision. The future: autonomous sterility assurance systems with real-time intelligence, a new era of reliability and control. When it comes to sterilization in place systems, several critical factors determine success. First, the system design has to be robust. It needs to handle real-world conditions without fail. Before starting, an effective cleaning in place process must be in place. Sterilization cycles must be validated thoroughly, ensuring complete steam penetration throughout. Biological indicators verify that sterilization is effective. Continuous monitoring keeps everything on track, while data integrity ensures trustworthy records. Finally, periodic requalification confirms ongoing compliance. All of this meets regulatory expectations for consistent sterility and product quality. To wrap up, sterilization in place, or SIP, is absolutely essential in sterile pharmaceutical manufacturing. It offers an automated, reliable way to ensure everything stays sterile, meeting strict GMP standards. By using validated cycles, strong equipment design, and constant monitoring, SIP protects product quality and patient safety. Looking ahead, as Pharma 4 evolves, smart SIP technologies will take this even further, boosting efficiency and compliance industry-wide. It's a cornerstone we can't do without. That brings us to the end. For any further support or questions, please don't hesitate to reach out. Thank you for your time and attention. It's been a pleasure sharing this with you today. This video was created with SlideSpeak. Visit SlideSpeak to turn your presentations into engaging AI-narrated videos.